Hi everybody. Today's lesson's on monocots and dicots. Yesterday we talked about angiosperms and how angiosperms, the flowering plants, are the most successful group of plants on this planet. They're successful because they have all the good adaptations of all the plants that came before them. They have vascular tissue, they have seeds, they have fruits, and they have flowers. Right? Which means today that most plants fall in this category of angiosperms. Over 75% of all plants that are found on this planet are angiosperms, which means that we have over 240,000 angiosperms. So because this group is so large, we can break it down into two smaller groups, the monocots and the dicots. Now, you can look at basically any part of the angiosperm and determine if it's a monocot or dicot as long as you know what to look for. For example, if you're looking at the seeds, in monocots, the seeds are going to exist as a single grain, right? Like a kernel of corn, right? It's just one solid piece. However, if you ever eaten something like peanuts, when you eat peanuts, sometimes they naturally just split into two halves. Well, that's a characteristic of a dicot. Now, inside of each of these seeds, monocots and dicots, you have the embryo. And monocots actually get their name because that embryo, that baby plant inside that seed, has what they call one cotyledon. A cotyledon is defined as the first leaves that are built into that embryo. So that first leaf that emerged from the seed. So in monocots, when this seed germinates, one little leaf will come poking out. In the dicot, di refers to two, and that's because you have two cotyledons, two of these embryonic seed leaves. So when this a seed germinates, you're going to have two little leaves coming out of that seed. But if the plant is already grown and you don't have seeds to look at, you can also look at the leaves of a fully grown plant. And you want to focus on the veins of the leaf. In a monocot, the veins are going to run perfectly parallel. Right? So they're going to go straight up and down this leaf. They're not going to intersect. They're not going to branch. They're going to be nice parallel veins. But in the dicot, the veins are more branched. They almost look like webbing here. So you have one major vein here, and then these minor veins kind of branching off of it. If you have a stem to look at, you don't really see the veins as clearly in the stem, but you do see the vascular tissue because it's always bundled up. So these vascular bundles, when you look at a stem under a microscope, in a monocot, they're going to be scattered throughout the stem. They're not really organized at all. But in the dicot, the vascular bundles are going to be all around the outside, or close to the outside of the stem, just inside the dermal tissue, and they're nice and organized and neat there, and they're arranged in kind of like a ring. If you're looking at flowers, you basically want to count the petals. If it's a monocot, the flower petals will always appear in multiples of 3. So 3, 6, 9 would be your monocot flowers. In dicots, the flower petals exist in multiples of 4 or 5. Now, the problem is that if you have a flower that has, a multiple of 12 or 15, you have to look at some other feature because 12 and 15 are both multiples of 3 and 4 or 5, like dicot. So, sometimes you can't just look at one characteristic alone and determine if it's a monocot or dicot. So the other characteristic that you could look at would be the roots. In monocots, they have something called a fibrous root system. So all the roots are about the same size here. There's no one major root that extends deep down into the soil. In dicots, they have what we call a taproot system. And that taproot system, you do see one main root that reaches far down in the soil, and then these small roots that kind of branch off of it. A carrot would be a good example of a taproot. Alright guys, that's going to be it for today's lesson. Thank you.